Ladies and gentlemen, the Cardiovascular Sciences Cutting Edge Technology Symposium is about to begin. We're delighted to express our heartfelt appreciation to our sponsoring partners, ATCG Limited, Beijing Yiran Technology Company Limited, Baoling, Miltenyi Biotech BV and Company KG, Shanghai Model Organism Center Incorporation, and all our value partners for their generous contributions that make this event possible. May I invite all the parties, please give them a warm round of applause to show appreciation. Thank you so much. Good morning, ladies and gentlemen. I'm Yuki, your MC for today. The Cardiovascular Sciences Cutting Edge Technology Symposium is happening. It's wonderful to see many talented young scientists and experts gather here today to share knowledge and innovation. Today's symposium is part of the Cultural Symposium and the Young Academy of Sciences Summit, YASS 2025 to 2027, organized by the Center for Excellence in Molecular Cell Science, Chinese Academy of Sciences, CUHK Joint Laboratory for Cardiovascular Sciences, the Cultural Foundation, and the Hong Kong Young Academy of Sciences, co-organized by the Tencent Charity Foundation with support from the Innovation and Technology Commission of the HKSAR government. The initiative showcases Hong Kong's strength as a global hub for science and technology. Now, let's officially begin today's event. Please join me in welcoming Professor Philip Chiu, Dean of Medicine at the Chinese University of Hong Kong, to deliver his opening remarks. Professor Chiu, please.
Mr. David Foster, Professor Kathy Lui, Professor Bin Zhou, distinguished guests, colleagues, young scientists, ladies and gentlemen, good morning. It's my great pleasure to be here representing CUHK and also Faculty of Medicine, the Chinese University of Hong Kong, in welcoming you all to the second Cultural Symposium and also the 7th subcommittee of the Young Academy of Science Summit, focusing on cardiovascular science and cutting-edge technologies. As a leading comprehensive research university in Asia, CUHK has always attached to great importance in contributing to the development of Hong Kong and our country through research, innovation, and cultivation and education of talents. We just discussed with David about the establishment of culture, which is very much tracing into the root of CUHK as well. So with the Faculty of Medicine, and across the border university, we are deeply committed to integrating research, innovation, and translational impactful outcomes through entrepreneurship. Our goal is to translate our research into tangible outcomes that tackle pressing global challenges and ultimately benefiting the mankind. This can be reflected currently with our global ranking, as CUHK Medicine has become one of the youngest top medical schools in just 45 years. To support this mission, the university has cultivated a robust global network of collaborators. We are home to approximately 300 research institutes and centers specializing in interdisciplinary research. CUHK proudly hosts 4 state key labs and maintain a rapidly growing network of satellites and research institutes in the dynamic Guangdong, Hong Kong, Macau, Greater Bay Area. Furthermore, CUHK is the host of 6 InnoCenters, out of which 4 are affiliated to CU Medicine, and leveraging on our local expertise and a wide network of prestigious global partners to generate translational impact on a local, national, and global scale. I'm actually one of the directors of this InnoCenter. It's a multi-scale medical robotics center. And in that perspective, we are also collaborating with global international partners, including Zurich ETH, Johns Hopkins, and Imperial College. And recently, we have in fact invited the ETH Zurich to come and test their system of magnetic guided robotic interventing guide wire for cardiovascular intervention. So in that perspective, it's very closely related to this symposium as well. In the medical and scientific field, bridging between the gap of bench to bedside is crucial. CHK has been a long-standing commitment to promoting collaboration between industry and university, as well as commercializing scientific research. So since 2014, the university have supported more than 180 startup companies and projects, some of which have been grown into very successful business, including 4 unicorns. So CHK, the Cultural Foundation, and the Hong Kong Young Academy of Sciences share a unified goal: promoting innovation and technology development in Hong Kong and facilitating interdisciplinary collaboration and knowledge exchange among scientists. This joint symposium offers a unique and invaluable platform for exchange of innovation, research ideas among our young academics, and to promote cross-disciplinary research collaboration. We are incredibly excited to have so many renowned young scientists and promising researchers gathering here today to share the scientific breakthroughs and new discovery in cardiovascular sciences. I look at the whole schedule. I'm also very excited to see the opportunity given by this summit to all the young academics and scientists to share vision and idea. And I think most importantly is the networking. I believe that we have to advance science together. So this is a great conference that will be highly stimulating and fruitful to all of us. Finally, I would like to express my best wishes to everyone here, and for the resounding success of the 2nd Culture Symposium and the 7th YSS Subconference on Cardiovascular Science and Cardiac Technology. Thank you so much.
Thank you, Professor Chiu, for the inspiring opening remark. Please be seated. Next, we are honored to invite our organizers to deliver their welcome messages. First, please join me in welcoming Mr. David Foster, Executive Director of the Croucher Foundation. Mr. Foster, please.
Professor Philip Choi, Professor Kathy Loy, Professor Bin Zhou, distinguished speakers, ladies and gentlemen, it's really my great honor to be here representing Croucher Foundation at the start of what looks like a terrifically exciting 2 days of talks here at CUHK. We were just talking about the origins of Croucher Foundation, and if it's okay, I'll just say a few words about that, because I think it's quite relevant to the discussions that you're going to be having going forward over the next 2 days. Croucher Foundation was set up in Hong Kong in 1979, and Philip mentioned that there was a very strong connection with CUHK because our founder, Mr. Croucher, was a very, very wealthy British businessman in Hong Kong who'd started work as a bellboy in a hotel in Macau at the age of 13. So he was not a scientist. He'd not even completed his secondary education, but he had become, in Hong Kong, extremely wealthy. There was a sadness in his life because he had a son, but they didn't get along very well. So towards the end of his life, he decided not to leave anything to his family, and he was then quite stuck. He didn't really know what to do. He didn't want to leave money to his family. He wanted to do something good for Hong Kong, but he didn't quite know what to do. The person who solved that problem for him was a chemist from the University of Cambridge called Alec Todd. He got the Nobel Prize in 1957. He was president of the Royal Society in the UK. He was master of Christ's College, Cambridge, and particularly relevant for these opening remarks, he was chairman of a foundation in the UK called the Nuffield Foundation, which does science funding. And the link with CUHK is that Lord Todd, in the very early years, was a council member of CUHK, and he was also the advisor who's on the advisory board for natural sciences for the university. So Lord Todd knew Hong Kong through Chinese University and through his participation in through his contribution to the development of CUHK in those early years, because just a few years after CUHK was set up in 1963. So just as Kathy and Bin Zhou met each other in Harvard, there was a very special meeting in Hong Kong in 1978 between Mr. Croucher, the person who had so much money but not really very clear idea about what he wanted to do, and Lord Todd, who had no money, but an extremely clear idea about what a science funding body like the Nuffield Foundation could do in Hong Kong. And it was quite an extraordinary idea because in those years, there were just two universities, CUHK, Hong Kong U, and there was no research funding at all. So you could learn to be an engineer or a doctor or a lawyer at a university in Hong Kong, but you couldn't really be a scientist because no research funding. So it was an extraordinary meeting and an extraordinary idea, and I can testify that Lord Todd was not only a great scientist, he got the Nobel Prize in 1957, but he was also a really good administrator. It's not often you have a combination of those two things. And I know that because when Noel Croucher asked him what the Croucher Foundation should do, how it would work, Lord Todd was staying in the Mandarin Oriental Hotel. That's a hotel on Hong Kong Island. He used the hotel notepaper. In pencil, he put some bullet points. He sketched out 1 page or 2 pages of bullet points. And then, since then, now is 2026, we simply follow these instructions. We haven't changed at all since Lord Todd set our direction. He particularly was keen that we should fund young people to do their research overseas. And Kathy is an example of somebody who won the Croucher postdoc fellowship to go to Harvard. Extremely tough competition. We just finished 60 interviews for this round of Croucher selection. So he was keen that we would support young people through competitive processes. But he was particularly keen that we would fund research through a peer review process and applying this standard of scientific excellence as judged through international peer review. So he launched that program 79, and we still use this program of peer review. And it was that competitive process that resulted in our funding Kathy's application for this conference, because that also is highly competitive. We received many applications for conferences like this one, and we chose to fund Kathy's conference not because she was a Croucher scholar, although of course we do know her, but because this gathering we felt was really important for Hong Kong, the topic, the way that Kathy and Bing Zhou had structured the event, and the way that CUHK and other funders like ITC, the Young Academy, are also willing to contribute. Feels like a genuine partnership. So I think that's all I want to say. I won't be able to stay because Croucher, one of the things that Croucher has also done since the very beginning, is one of Lord Todd's bullet points, is to introduce science to very young people in Hong Kong. In this case, primary school students. We have a festival called Croucher Science Week. There are more than 90 events. It's a free festival. There are people from the Deutsche Museum in Germany coming, from the London Science Museum, the Royal Institution, from Singapore, from Spain, coming to perform science shows for young people in a bilingual setting because we have an interpreter. So we usually have about 30,000 Hong Kong primary school students come to that Croucher Science Week, and it's starting next week. I cannot advertise it to you because all of those events, 90 events, are sold out. They sold out within 8 hours of launching our website. And in fact, the website crashed. So the Croucher Science Week turns out to be so popular that we're going to have to, next year, we're going to have to maybe have even more than 90 events, I think. So I just want to say, I hope that I know the discussions will go well. I'll wait to get the report from Kathy and Bin Zhou, and we'll see if we funded one event 2 years ago. I hope that we can fund again in 2028.
Thank you, Mr. Foster, for the story. Please be seated. It's very interesting and impressive at the same time. Next, we are honored to welcome Professor Kathy Lui, co-chair of the event, Croucher Scholar, professor in the Department of Chemical Pathology at the Chinese University of Hong Kong, and the founding member of the Hong Kong Young Academy of Sciences, to deliver her welcome remarks. Please join me in welcoming Professor Lui.
So, distinguished guests, Professor Chiu, Mr. Foster, Professor Zhou, all our colleagues and friends, welcome to Hong Kong. And also thank you so much for your support to this symposium. This is actually the second time we organize the symposium in cardiovascular sciences. So with this opportunity, I would also want to thank our speakers, especially those coming from afar. And also, all of your support will be very valuable as the foundation for our discussion, future collaborations. I would also want to thank our organizing committee members and also all the funding agencies that make this event possible. This event was actually prepared for a long time, especially by the secretariats of ASHA, the Academy of Sciences of Hong Kong, especially Ada, Oscar and Anton, and also my teams who offer some help for the speakers, and also CK who has communicating with lots of speakers. So thank you so much for your support. I feel so guilty compared to you guys as I didn't put as much efforts as you, but I'm sure like this is a teamwork that we work together to make the conference meaningful. I want to also introduce this one of the most important support from the Croucher Foundation to establish the CAS-CUHK joint laboratories for cardiovascular sciences, which based on actually decade-long of collaborations as Bin and I met in Harvard. Also, I was a Croucher scholar. So since that time, we collaborate a lot in this area, and also we feel that this is very important research field that we want to develop in Hong Kong. So we hope to, through like not only both of our labs, but also connecting with the world. So 2 years ago, we organized the first Croucher Symposium in cardiovascular sciences, and also the Young Academy of Science of Hong Kong focusing on regenerative medicine. So we decided to combine this into one event this year. As David said, we also hope to keep this continuous, like we can do it every 2 years. So we hope that we can also welcome you and also have your kind support 2 years later in getting more leading experts from abroad to come to Hong Kong and develop this area globally. So I also want to take this opportunity to introduce the Young Academy of Sciences for Hong Kong. It is actually a young chapter of the Academy of Sciences of Hong Kong, which has established 10 years ago. So one of the very important mission, which also like is similar to just like David said from CHK, and also what David said from Croucher Foundation, or Philip said from CHK, is to really nurture the development of science in Hong Kong. So there are a few missions for us, including fostering scientific excellence, developing Hong Kong as a innovation and technology hub, and also advancing scientific education. So we put a lot of effort in primary and also secondary school to promote science, because it is not their first choice in Korea. So we hope to bring more scientists in Hong Kong and nurture them since young. And also we also inform the government through policy advisory. So these are the major missions of the our academy, and we hope that we will grow. So this year we celebrate the 10th anniversary, and as you see, we have a lot more young academics joining us. So today's event is one of the events that is also correlate with what CHK, YSH, and Croucher wants us to put together. And also we chose a topic, cardiovascular sciences, because it is actually the most important, one of the most important diseases, and also the leading cause of deaths in the past 3 decades. And especially in China, around 50% of all deaths are correlated to cardiovascular sciences. Cardiovascular disease, but a lot has not been known. So today we will cover a range of topics, including both the cardiac and also the vascular side, and we also will go into the causes in terms of like genetics or epigenetics courses and to study the disease better. So another major part of our conference focus on the to encourage and also engage the next generation of young scientists. So tomorrow there will be a section mainly presented by our trainees, including postdocs and PhD students, to introduce their work. And also around the corner, you will also see a lot of posters that prepared by our trainees. So we also hope to engage the collaborations not only within the PIs, but also some new stimulating discussion between our trainees and the leaders from the world. So we hope that this conference will give all of us some meaningful gifts. And most importantly, we also want to introduce that CHK is actually the largest university in Hong Kong. We have many beautiful green areas. If you have time and if you have chance, I hope that you can also go around our campus. And last but not least, we hope to welcome all of you to Hong Kong and also to especially those from abroad. And we also hope that the conference will give you a meaningful memory in the years to come. So thank you all of you for participating in this conference.
Thank you, Professor Lui. Please remain on stage. Please remain on stage. Thank you very much for your sharing. May I invite the following guests to join Professor Lui for a photo. They are Professor Philip Chiu, a round of applause. Professor Zhou Bing. Mr. David Foster. Continue the applause. Professor Huang Yu. Professor Stephanie Demola. Professor Thomas Brown. Professor Ralph Adams, Dr. Yevgenia Nuzinovic, Professor Wang Yibing, Professor Helmut Augustin, and Professor Sean Wu. Please join us on stage for a group photo. Okay, let's look at our official photographer. Big smile. Yep, big smile. And promote science and technology to our young generations. Okay, give a like. Thumbs up, thumbs up. Yes, yes. Promotion photos. One more. Thank you so much. Thank you so much. With Professor Philip Chiu, Professor Kevin Lui, Professor Zhou Bing, and Mr. David Foster, please remain on stage. For the other guests, please be seated. And may I continue to invite the following guests to come on stage for photo. They are Professor Randolph Wong, Professor Xu Qingbo, Professor Nicholas Smart, Dr. Nie Yu, Professor Zhao Hui, Professor Zhang Li, Professor Ronald Wang, Professor Wei Ho Tang, Dr. Owen Ko, Dr. Tian Xiu Yu, and Dr. Alan Poon. A warm welcome from every part of the world, and thank you for joining us today. So let's look at our photographer in the center. It's definitely a representation of excellence, and I'm very sure we will learn a lot, get insights, and exchange a lot of new ideas. Okay, thumbs up. With the smile as well. All right, brilliant. Thank you so much. With Professor Chiu, Professor Kevin Lui, Professor Zhou Bing, and Mr. David Foster, please continue to remain on stage. May I now invite the following guests to also come on the stage to join us for the group photo. They are Dr. Zhang Mingjun, plus Mr. Liu Zitao, Mr. Hou Yangfeng, Ms. Li Yangxian, Dr. Luo Minghao, Dr. You Zehao, Dr. Hong Hui Ling, Mr. Muladini Ahmati, Mr. Xuan Zong Su, Mr. Fan Dongxiao, and Ms. Li Jie. Last but not the least, Ms. Shi Mengsha. A round of applause. All right, let's look at our photographer. And we look forward to hearing your findings and research so that we can always keep improving each day. Thumbs up is an automatic one. Yeah, you know your role. Okay, make sure we look at the photographer and everybody thumbs up and look at, yes. Enthusiasm. All right. Thank you so much. Everyone, please return to your seat. Thank you so much. And we will let the photographer take a group picture for everybody. So, yeah, so let's come together. Yes, we can come closer together for a group picture. We try to more centered to the stage. Yes. Okay. So while our photographer is preparing, for this side of the audience, can you slightly move to this side for a while and then can move back later? Thank you very much for your cooperation. While we are tidying up, I mean, ladies and gentlemen, you can tidy your hair and put some lipstick on, or just tidy the mustache for the best effect of the photo. I know it's early in the morning, so let's give some enthusiasm for your smiles. And later on, the last picture, we will also do a thumbs up as well to boost up the energy. 3, 2, 1. 3, 2, 1. Close up. We now move to session 2, vascular disease, and we are privileged to have Professor Alex Lee from the Department of Medicine and Therapeutics and Professor Fei Zhang from the School of Life Sciences, both at the Chinese University of Hong Kong, serving as the moderator for this session. May I now welcome Professor Lee and Professor Zhang to the stage to lead us through this exciting session. Thank you very much.
Okay. Hello. So, good morning, everyone. So the second section of today will be on vascular disease. So I'm Fei Zhang from School of Life Science, and next to me is Professor Alex Lee. These sections, we will have several international well-known speakers to you. So the first speaker of today would be Professor Stephanie Damila. So she is the Director of Institute of Cardiovascular Regeneration from University of Frankfurt. So her talk of today will be on senescence and vascular crosstalk as drivers and therapeutic targets of cardiac aging. So without further ado, may we welcome Professor Damila for the talk.
So thank you very much, first of all, to the organizers, Kathy and Bin, for this wonderful invitation, and also the entire team for the hospitality. And it's a wonderful occasion to be here in Hong Kong and to be invited to this meeting. My talk will now focus on aging, and of course, aging is interesting for all of us because everybody is affected by aging. But I think in the cardiovascular field, it's particularly interesting because we have dealt well with the major risk factor for cardiovascular disease. We can treat hypercholesteremia, hypertension, mainly also obesity, but aging remains one of the so far unmodifiable risk factor, which may account for many of the effects we have seen attributed to aging and cardiovascular diseases. Aging also promotes heart failure, not only because it increases acute myocardial infarction and atherosclerosis, but also because age and also obesity contribute to heart failure with preserved ejection fraction. Heart failure with preserved ejection fraction is a different type of heart failure where the heart is stiffening and therefore can no longer pump adequately blood to the body So you also have a reduction in oxygenation, but you have a different pathomechanism, which is mainly due to fibrosis, as said, and diastolic dysfunction. So we were interested in the contribution of aging to cardiovascular disease, particularly in the heart, and we're assessing the interaction of cells in the heart beyond the cardiomyocytes. Of course, the vascular cells, immune cells, and also the fibroblasts and bone marrow-derived immune cells coming into the heart and being increased during aging contribute to age-induced dysfunction of the heart. And this project was led by Julian Wagner, and who did the wet lab part, and David Rodriguez Morales, who is responsible for the bioinformatic part mainly. And we are trying to understand how aging affects cardiovascular disease and heart function by now addressing the trajectory of aging. While we don't have all the data ready yet, the data today I will present are focusing on a quite early type of aging. This is very important because at different stages of aging you may have different changes in the heart function and also in the contribution of cells to dysfunction. And this is the part, so our mice are mainly 18 months old. This resembles about my age. So starting to address the, yeah, we are affected, yeah. So when we did so, we first assessed used spatial transcriptomics to assess the trajectory of aging, and we found surprisingly, and I won't show too much data, but we found that the vascular niche in the heart is one of the first regions where we found increased senescence and increased aging signatures, suggesting that the vasculature may be a prime site. And actually at the same time, when we published this work in mice, there is also a Cell paper which showed actually in humans that also in this trajectory analysis, blood vessels were shown to be the first tissue that ages, suggesting that blood vessels may have a really instigating role early in aging. And it's maybe not surprising if you plate one human's endothelial cells, for example, that this may have a huge impact on your systemic also response and paracrine factors all over the body. You have vessels in all organs, so this may have an effect, of course, on aging. So therefore, we were particularly interested in studying the vasculature and endothelial cells in the heart. And this just summarizes the functions, of course, beyond providing nutrients and oxygen to the tissue. Endothelial cells and lymphatic vessels are also important for waste export, something which is understudied so far. They can interact with fibroblasts, as have shown by several of ours, but also other papers. Endothelial cells have a critical role in regulating immune access and also export. And of course, they control also vascular function and pericyte and smooth muscle cells are important to control the diameter of the vessels. To get some molecular insights what aging is affecting, how aging is affecting endothelial cells, we use single cell or single nuclear RNA sequencing to start with and explore the effects of aging in the mouse heart, comparing 80 months to young mice hearts. And beyond the things I've just shown you, that there are a lot of factors dysregulated, we made one surprising factor. We found one surprising transcriptional profile which I would like to share with you, namely that we found significantly upregulated genes in old endothelial cells related to axon injury and neuron cell death. And we were very surprised by this finding because we were not having this into our minds that endothelial cells and nerves may interact in aging. But if you go back, nerves and vessels co-develop. And if you see here in this immunostaining, nerves in green here, and in pinkish, the smooth muscles actin, positive vessels, and also the capillaries in IB4, there's a close interaction. And if you look into more details, the different parts of the vessels have different nerves, but all of them are closely in proximity to the vasculature. So sensory, sympathetic, or also parasympathetic nerves are in close proximity. So we wonder whether aging may affect this neurovascular interface, and maybe this could contribute also to age-associated dysfunction. And when doing so, we first assessed the nerves. So these are the axons in the heart. These are mouse hearts. The nerve bodies are outside, and they project into the heart. And you can easily see from this staining that compared to young mice hearts, old mice have had less axon density. So the axons are reduced, and this was related to all the different type of nerves we have studied also in detail. And this happens quite early. I mentioned the 16-8 month window. At this time, already the nerves decline. So what is the mechanism? It turned out that there is a profound change in the gene expression in endothelial cells. So there is an induction of so-called repulsive signals. These are signals which can lead to the axon regression of the heart. And there is also a decline in neuroprotective signals. And I brought you two examples for repulsive signals. This is semaphorine 3A, which is induced in old heart endothelial cells. If you treat, for example, cardiac organoids, you see here the decline in nerve density, as you have seen the staining here for TUCI1. This is a nerve marker. This is declined. And also in vivo, if we overexpress semaphorine 3A, particularly in endothelial cells, you can appreciate the decline in nerve density in the heart. On the other hand, we also have a decline in neuroprotective factors. VEGF-B is one of the factors which is important for nerve growth. This is declined in the old heart endothelial cells. And if we overexpress VEGF-B together with cariaditalo, we found indeed that we could restore also some of these innervation defects, suggesting that there is a close crosstalk between endothelial cells and nerves in the aging heart. So we believe that we should add the innervation to the hallmarks of aging in the heart. And of course, the question is next, what is the upstream regulator? So what processes drive this denervation? Of course, aging has multiple effects. You see here the hallmarks of aging. And we were focusing on senescence. So you may know senescence has a double-edged role. So I have to go back here. Has a double-edged role. It's very important for injury response, as we have seen by Bin Shu's data, but also by Eldad Zahor, showing that it's important in the repair of, for example, the heart or the liver. This is acutely. But if you have senescence chronically, senescent cells can release so-called senescence-associated secretome protein, and this may have really detrimental effects on tissue homeostasis. And we were inspired by our finding that this repulsive signal, semaphorine 3A, was increased in long-term Paget's endothelial cells, suggesting that it may be part of this senescence-associated secretome, and asked the questions whether senescence may be the upstream regulator to induce denervation. So to address this question, we used so-called senolytics. Senolytics are drugs which can kill senescent cells, and one of the first generation senolytics which we used was dasatinib and quercetin here. And we treated mice, old mice, either with placebo or with these senolytics, and then assessed the effects on the heart. And you see here in young hearts, this is cellular senescence with acidic beta-gal staining. You see here in old hearts more senescence. If you treat the mice with senolytics, there's a reduction of senescent cells. At the same time, you can appreciate that the nerves, these are in green, the TUCI1 staining, a decline in old, and also restoration by the senolytic treatment, suggesting that indeed senescence somehow is really an upstream regulator of this denervation. So what is the consequences of all of this? Can we restore functional consequences of this denervation? And this is quite difficult to address because we would now measure nerve functions, but this is on the way. But what we did, we measured heart rate variability, which is controlled also by the innervation. And you can appreciate here that the decline in heart rate variability can be restored by senolytics. And it's very interesting, you see here in young mice, even the day-night cycle, in young mice, it's very nicely up in day and low in night. This is no longer true in the old mice, where they have a lack of this regulation. And in senolytics, we can restore this to some extent. This may be particularly important because we also know that elderly have a reduction in this heart rate variability. Second, we measured electrical stability. We know that denervation can lead to silent ischemia and lethal arrhythmias. And also here we found an increase in inducible arrhythmias in old mice, which was restored by senolytic treatment. Finally, we looked into functional consequences of this senolytic treatment. And as I mentioned, one of the hallmarks of aging is diastolic dysfunction. You see here the increase in diastolic dysfunction in 18-month-old mice, and this was reduced by the DQ senolytic treatment, and also fibrosis was to some extent reduced if you treat the mice here for these 2 months. Together, these data suggest that in the aging heart, we have a dysbalance of axon repulsive and guidance and protective factors. This leads to a regression of axons, and we can restore this by senolytic treatment. In addition, I will not have time to follow this up, but we have additional data that particularly in these vascular niches, which are the first to age, we have also a dysbalance of macrophages with increased bone-derived macrophages and a decrease in tissue-resident protective macrophages. So we suggest currently and investigate how these macrophages and the clearance of senescent cells may be dysregulated in aging, and we propose that maybe really a neurovascular immune crosstalk in these niches in the aging heart. So this is more like a teaser. I hope I can present next time if I hopefully be invited more about this work. But finally, in my last 5 minutes, I would like to discuss how we may use senolytic treatment or what would be alternative options. We were very impressed, I must say, by these senolytic treatment effects, but one has to be taking into account that this may need a very sweet timing, because you can appreciate if you have a few senescent cells, this would not work. If you have a very high number of senescent cells, this may be detrimental, because if you kill too many senescent cells, you may really ruin the remaining homeostasis of the tissue. So there is likely a sweet spot for the treatment with senolytics. Second, you may need repair capacity. If you kill the senescent cells and there is no repair capacity, this will not be good for the organ. So we believe that we may need really a biomarker-guided therapy and maybe short-term windows to clean senescent cells at a relatively early time to make this successful. This is not so easy because you have to convince someone like me to take maybe the senolytic treatment, and you will not be maybe successful if you take an 80 or 90-year-old. So this will be a challenge for the field in my view. And second, of course, one could think whether we find alternative strategies to more specifically interfere with pathways of the senescence-associated secretomes or the functional dysfunction of the cells. So as said in the last few minutes, I would like to talk with you about alternative strategies. And we asked the questions, what happens to endothelial cells in the aging heart? Could there be epigenetic dysregulation which lead to a long-term dysfunction of endothelial cells? And maybe we find some genes which are repressed, which may be upstream regulators or activated, which may be upstream regulators of endothelial cell aging. And actually, together with Thomas Braun, we looked into single-cell attack sequencing quite early on when the technology was established in Thomas' lab. And we found there's one transcription factor called zinc finger and PDB domain-containing protein 16, which was strongly repressed in aging. And this transcription factor, as said, there was repressed, so the chromatin is closed. And consistently, we found that there is a reduction of this expression on mRNA level. You see here a single nuclear RNA sequencing data in humans and in mice. There is a decline of the expression in endothelial cells, but also in the total heart protein, you see also reduction of the protein and suggesting that there is a reduction of this transcription factor. What is this transcription factor doing? So there is not so much known, but if you have homozygote mice deficient in ZBTB16, they also have defects. So there are some mice are also dying. They have also skeletal skeletal defects. So we used heterozygote mice to get some first insights because they were viable and had no obvious defects. And we wonder what is the function in the heart. So there is a half maximal reduction in endothelial cells in these heterozygote mice. And we were very interested to see that there is an increase in diastolic dysfunction in the heterozygote mice. There's an increase in LV mass and also hypertrophy, cardiomyocyte size is increasing, but no change in systolic function. So this is clearly like an age-associated effect. And interstitial fibrosis and senescence was also increased. Of course, these are systemic dysregulation of this transcription factor. So we wonder what happens if we only deplete this in endothelial cells. So we generated endothelial-specific knockout mice. And actually, I have not all the data here, but just show the diastolic dysfunction, which was very similarly affected, suggesting that this transcription factor really has a key role in endothelial cell aging and also in inducing diastolic dysfunction by an endothelial cell manipulation, which is not inducing ischemia. So we wonder what is the upstream mechanisms and what are the regulated genes induced by this transcription factor. And we found that a surprising large number of genes being associated with secreted factors were regulated if we knocked out ZBTB16 in the endothelial cells. So we wonder whether there might be a paracrine effect which may explain the findings we have seen. And we used the supernatants of these siRNA-treated cells and added them, for example, to fibroblasts to see whether this might indeed induce fibroblast activation. And we could see that, for example, collagen expression in these fibroblasts were increased. Also, if we used organoids to be treated with these supernatants, there's also an increase in collagen expression and smooth muscle actin expression, suggesting that there's really a crosstalk between the two cell types, at least in vitro. We also assessed the effects on cardiomyocytes by doing co-cultures. You see here these ZBTB16 endothelial cells. They are not as nice as the controls. In this V-Caterian staining, you see that the cells are no longer as functional active and the morphology changes. But the cardiomyocytes really also reacted to this. You see here a hypertrophy of cardiomyocytes here in green, suggesting that there is also a paracrine crosstalk between them. We could measure the beating frequency of the cardiomyocytes. This is a typical stress response of cardiomyocytes contraction, and also an increase, a statistical increase in hypertrophic responses, demonstrating that also in this crosstalk is an effect on cardiomyocytes, which may explain also the diastolic dysfunction we are seeing. Finally, suggesting that this transcription factor controls endothelial cells, but has important effects on the neighboring cells in the heart, so induces fibroblast activation, cardiomyocyte hypertrophy. And of course, we also looked into axon density because this was, as I have shown you, our preliminary findings. And we also found that there is a reduction in neural sprouting. With this, we conclude that this transcription factor may be really a critical regulator of endothelial cell function, and wonder if we can restore vascular function and also the cardiac function when we overexpress ZBTB16 in endothelial cells. And we did so by using an endothelial cell targeting strategy, which were provided by a collaboration with Oliver Müller and Christian Kuppert. And you can appreciate here that this is really targeting nicely the endothelial cells, this viruses, while the non-endothelial fraction is less targeted. And you can appreciate that the overexpression of ZBTB16 reduced diastolic dysfunction, and it also had an effect on vascular fibrosis. You can see here the 18 months and 20 months data with the control. This remains. The overexpression of the AAV at 18 months restored fibrosis to 18 months level, but it did not reverse. This suggests that we cannot revert a phenotype, but rather prevent secondary fibrosis, which is consistent with a paracrine activation. With this, I would like to conclude, and I hope I've convinced you that senescence, although it's a complicated reparative and also detrimental response, during aging, it certainly has a detrimental function by chronically releasing paracrine activity. We have shown an epigenetic control of gene expression, a repression of this transcription factor in endothelial cells. We have found an altered paracrine activity of the vasculature and a control of fibroblast and cardiomyocyte function indirectly by endothelial cells, which propose that we may have an interacting strategy also to prevent the further onsets of age-associated processes. With this, I would like to conclude. Thank all the people in the group. It's really a team effort and the funding agencies. And I would like to highlight again the contribution of Julian Wagner. He has now his own research group, and he was really the driver of all these age projects, and a number of people from the lab, including Katrin Stiltz for the ZBTB16 story, Veronica Indenada for working on the senescence vascular niche, David, I mentioned before, for the bioinformatics, but also the entire bioinformatic team who was necessary to support the data, and many collaborators, which I have already alluded to, and thank again here at the end. Thank you very much for your attention.
Okay.
Thank you, Dr. Stephanie. Is there any other question? Yes.
Hi, Stephanie. Very beautiful work. I'm wondering, in your SEMA 3A work, if you knock out the SEMA 3A, will this change, improve the neuron endothelial interaction and also change the SASP of endothelial cell in the aging model?
So we haven't done the experiment so far. The problem is knocking out SEMA4A must be really endothelial cell specific, because if you have a general knockout, you have really detrimental effects also.
Yeah.
And we didn't do the experiment yet with the short hairpin, because the short hairpins were not as effective at the time when we tested it, so we would have to develop them further. And the knockouts they are still breeding, so you need 20 months. But I can't tell answer.
Okay, thank you.
Stephanie, thank you so much. Fantastic. 2 questions. One is people in Germany, do they use quercetin? You know, you mentioned you use it for animals. That's from Chinese herb. Quercetin, yes. The health product. Yeah. Do people use it in Germany? Or?
I know that you can buy it in the pharmacy.
Yes.
I must say, if you look up the concentrations and the quercetin alone, didn't do anything in our models. So maybe you need the appropriate concentration. Mm-hmm. And the dasatinib was, at least in our hands, essential in combination to kill the senescent cells. Yes. Quercetin is, of course, very important antioxidants. And I must say, with the senolytic approach, it's really a combination of powerful drugs. Right. And it can maybe, can affect maybe also other pathways.
My real question is, any change to the heart rate in the aged animals, if there's a loss of autonomic nerve, sensory nerve, for example?
So certainly we have seen the regeneration of the nerves, but I think that this is likely the case. But of course, heart variability is also a complex readout, and we need to measure specific the nerve function with more appropriate technologies, and this we are currently doing. The aging work is very long term, So as said, I don't have the direct measurements. We don't have yet. Yeah.
Would you think the sensory nerve might be beneficial in a sense when they release substance P, CGRP, for example, increase endothelial cell function?
Certainly the nerves are, let's say that the sensory nerves are certainly beneficial, but also the sympathetic, that let's say that it may be very important that you have also the spatial distribution of nerves may be very important. And if you have a lack of nerves in one region of the heart, and then relative increase in the nerve density in the other part, this may be also not so healthy, even for sympathetic nerves. You know what I mean? It's like it's not only for heart variability in the sinus node, but also across the ventricle. It's not so well understood what the sympathetic nerves actually are really doing, and the dysbalance may also be detrimental.
Can I ask you a question? I'm a cardiologist, and thank you very much for your excellent talk on this, especially about the relationship with aging and with the neurologists. It's fascinating. So my question is there any drugs being used now in the clinical setting for HFpEF, for example, SGLT2 inhibitors and mineraloid receptor? Are they have some effect on the nerves itself, or it's in a different pathway?
So SGLT2 inhibitors at least reduce senescence, and with our data you would assume that there is a change also in nerve density, but we haven't done the experiment, so we have to do it.
Okay. Stephanie, Javier, the aging leads to VEGF signaling insufficiency and vascular rarefaction putatively through dysregulated VEGF receptor 1 soluble. Now, would you think that chronic delivery of senolytics can do the same, as Elie Cachette has shown that low-dose VEGF overexpression ameliorates the effects of aging, of vascular aging? Mm-hmm. In other words, the question I'm asking is, what is bottleneck for vascular aging? The appearance of individual senolytic cells that you can remove, or the aging of the population of cells?
So the point is, at the 18 months, we don't see capillary rarefaction. I didn't show you the data. It comes later. So the denervation and senescence comes a few months earlier than the decline in capillary density, which is very surprising, but we showed this now in humans and in mice, and with many mice in both sexes. So my assumption is that this decline in endothelial cell numbers comes later. This is the first point. And these experiments were done at a time where endothelial cells still have the capacity to repair and regenerate, being proliferative. Still, I would say long-term treatment of senolytics may not be a good thing. So I think you would need to have this like a fasting cure, like a few weeks, and then you should stop it. I don't believe that it's good to have this chronically depleted. And it's a very interesting question. VEGF signaling, we see high levels of VEGF, also on protein, VEGF-A, but the signaling is dysfunctional. So there is really, it's not the growth factor, it's like the signaling which is somehow impaired in endothelial cells. So this is an interesting additional study. I don't know whether this links to senescence.
Did you check this is free VEGF or complex VEGF?
We did not do the details, but it's at least it's there, and we didn't look at the isoforms, but we see the protein and we see the RNA quite highly expressed, even higher than at baseline.
Hi, I enjoyed your talk very much. Thank you. Regarding ZBTV16, I wasn't clear, how do you think it actually activates the fibroblasts? I mean, is it through a process of changing their identity, or how does that work?
So the story is not yet fully completed, but we suggest that it's a PDGF related pathway which is dysregulated by ZBTB16. Also here we have follow-up studies going on, and that this crosstalk is somehow affected.
So in ZBTB16 knockout mice, is there a problem with their fibroblasts?
So certainly we have more fibroblast activation, so collagen expression is increased and we have more fibrosis, yes.
So because of the time, so thank you very much for Professor Damila's work. So let's move to the next speaker. The next speaker of these sections would be Professor Yu Wang. So Professor Wang is the head and chair professor of biomedical sciences and vascular biology in Jean Hu Professorship in Biomedical Sciences from the City University of Hong Kong. So Professor Wang today will be talking to us vasoprotective roles of uncoupling protein 2. So welcome.
Good morning, Hui Zheng. I would like to thank the Kathy and also Zhou Bing for organizing this joint symposium. Now, these are excellent presentation of previous 5 speakers, and further convinced me. And I have chosen a good topic, focus for my career on the endothelial cells. And the endothelial cell actually is the key to the homeostasis to all organs, bone, liver, for example, today, and the heart. And of course, I will study mainly focus on blood vessels. I've been studying the endothelial function, dysfunction in animal models for many years. And basically, in endothelial cells, there's a very well regulated balance between nitric oxide, and which is simple, derived from enos, and also the reactive the oxygen species, which actually produced by a bunch of those enzymes. Now, this is very important. If you maintain this balance, and your endothelial cell should be healthy. And the plus, of course, today we're talking about a little bit physical activity, which I don't have time to discuss today. Now, under pathological conditions, for example, hypertension, dyslipidemia, diabetes, for example, now partly due to loss of nitric oxide, which normally control actually the level of endothelial cell ROS. Now, the ROS actually outperform nitric oxide and induce the oxidative stress. Those actually is the key to the vascular dysfunction under many of those conditions. And therapeutically, of course, we try to restore the balance which is disrupted under pathological condition. So any drugs, actually, you try to either suppress the ROS generation or directly or indirectly increase the nitric oxide bioavailability will help to restore the endothelial function under those conditions. Now, nitric oxide, as I mentioned, is a gas molecule, and then derived from the L-arginine. But the RS actually in our endothelial cell, or virtually all the mammalian cells, there are many of those enzymes contribute to the elevation of free radicals. And today, actually, I will focus on one particular source, mitochondrial RS. And commonly in the cardiovascular system, we often use angiotensin II, for example, to trigger the induce the blood pressure. And actually, this is a very powerful inducer of NADPH oxidase. There's so many of those enzymes able to induce RS, including nitric oxide synthesis. And when they uncoupled, for example, release free radical instead of nitric oxide. Now, same time, now all these RS can be balanced by a very powerful antioxidant defensive mechanism. Top three here certainly is the most important in all the cells. The SOD, that we study quite a lot, the catalyst, for example, and the others. Now, what is the uncoupling protein 2, UCP2? Now, UCP2 actually localized in the inner membrane of mitochondria, which actually is a pump, the proton, the transporter, and expressed by all endothelial, the vascular cells, including endothelial cell, muscle cells, and the macrophage, for example, in the heart as well. Now, which actually is a very potent controller or regulator of RS, and the mitochondria probably provide a major source of free radicals. And this actually we've been interested for some years. And because RS is very dynamic, very difficult to capture at a certain stage. So currently we try to focus on how these things, particularly inside the cell, different organisms, for example, ERs, for example, they might interact with mitochondria and control overall the RS level in those cells. Now, I just give you briefly two examples we study on the role of endothelial UCP2 in maintaining endothelial function. The first one is the diabetes, because there's actually led by the Tian Xiaoyu. That time, she was my PhD student in this university. And we knock out, that time we did not have much tool, yeah? We knock out, global knockout UCP2. And then you can see here, now use acetylcholine to stimulate endothelial cell release nitric oxide as an index of the condition of endothelial function. So here you can see very clearly, now in the knockout UCP2 knockout mice, and the acetylcholine induced relaxation, a little bit impaired, not that much, yeah? And this impaired relaxation can be brought back by acutely by those the RS scavenger, including microQ, which is supposed to be relatively selective to remove mitochondrial-derived RS. Now, in another type of experiment, what we dissect those the piece of artery, in this case, proper aorta, and then you get exposed to the high glucose. You can see obvious impairment in the acetylcholine-induced relaxation. Now, all these can be reversed by the mitoQ, for example, which is suppress mitochondrial RS. We also measure the mitochondrial-derived RS. Now, you use those the relatively specific the diet, and these are very clear. In this case, we use the LPC, for example, able to trigger mitochondrial RS generation. All these can be suppressed now by the RS scavenger. For small summary, because here you can see now if you knock out the UCP2, and then you can see there's obvious enhancement in the acetylcholine-induced impairment. Now, in response to, for example, high glucose, and which can be reversed almost totally, yeah, by the RS scavenger. including the scavenger for the endothelium-derived RS. And also we expose, for example, in the diet-induced obese mice, and you can see there's impairment in the acetylcholine-induced relaxation, which can be rescued as well when you overexpress UCP2 in the vascular tissue. Now, does UCP2 contribute to vascular benefit of any drugs that improve endothelial function? And at the time, actually, we did not know. So we studied one compound just to show you, sitagliptin. Probably now you know sitagliptin actually is, not now, but in a couple of years ago, quite widely used. Try to increase the availability of the GLP-1, because the GLP-1 actually can be degraded by DPP-4. And these drugs, because at that time, there's very limited the drugs available which can act as a selective GLP-1 receptor agonist, for example. And then we got these compounds, and then we treat the animal. In this case, we work on both hypertensive and the diabetic rodents. But it did just give you one example. But in this case, what we did, in the hypertensive rats, spontaneously hypertensive rats, if you suppress enos activity, for example, you just use L9, for example, which the L-arginine analog competing for the activity of the enos, and then you add acetylcholine. In this case, instead of cause relaxation, actually cause contraction. And then we study quite for long years. We know what is a mediator. Actually, those are COX-2 here, derived for some PGI2 or other prostaglandin, which are acting on muscle cell cause contraction. Now, in this case, you can see very clearly it is a chronic 2-week treatment. Now, with the sitagliptin, which is able to lower blood pressure a little bit. And then here you can see very potently now suppress acetylcholine-induced contraction. This is another way to study the endothelial dysfunction in animal models. Now, do not need to bother you much. And this is go through this the MPK or cyclin AMP MPK system. But in the end, of course, if you use this GLP, this sitagliptin, for example, they will increase now local production of GLP-1, and then will increase UCP2. This is probably the first study we show at least GLP-1 agonists, able to increase UCP2 in the vascular, the wall. Now, and also also we found now what is the cause this increase RS, increase the COX-2 expression, because mainly it's RS, because we spent quite a number of years to study, because all those conditions which can increase now endothelial natural the RS generation now through this the ERK pathway, for example, then increase expression of COX-2, then release those PGF2 alpha, for example, in our case, and the cause contraction. Now, and in this case, we use a pharmacological inhibitor of all different steps, for example, leading to activation of COX-2 or expression of COX-2, now able to normalize, for example, now all those changes in the vascular wall. And functionally, of course, because this is repeating some of our early studies. Now, all these as I mentioned, RS is the key to the expression of COX-2 in the vascular wall. Now, if you inhibit the RS, for example, use RS scavenger, and this is quite effective to suppress now endothelium dependent the vasocontraction. Again, we use quite a lot of these pharmacological agents, and those are very useful to our study. And I will skip this one. Now, also, we're able to get a piece of renal artery from hypertensive patient from local hospital. Now, and here is very clearly, actually, just like in the renal artery from spontaneously hypertensive rest. Now, this renal artery from hypertensive patient also, they express high level of COX-2 and low level of UCP2. Now, if you treat now this piece of tissue with GLP-1 receptor agonist, in this case, extension 4, for example, now you can see very obvious the beneficial effect. Now, bring back this lost UCP2 and suppress COX-2, for example. And functionally, of course, we can see significantly improve endothelial function. So I'm not going to bore you, but this is quite interesting, and we continue to study, but of course now we study semaglutide, not study extension 4 anymore, and we study on another topic, for example, they're also acting on UCP2 in the brain endothelial cells, for example, how they're going to control the integrity of the endothelial cell barrier. Now, last bit of my talk about, because this is unexpected, and then because this couple of years we spent quite a lot of time on the study impact of these shear forces, either laminar flow, distal flow, for example, now in controlling endothelial function, particularly related to the atherogenesis. Now here is out of the RNA sequencing data, we found UCP2. Mitochondrial UCP2 actually is mechanosensitive, of course, because this UCP2 is not transcriptional factor. They must be controlled by something else. But at least, it really is the mechanosensitive. And we find laminar flow now not only in the animal, in the arteries, for example, which is experience this different type of flow. You can see the different expression level in those the sports. And I give you a little bit. Yeah. Now in this ABIDI system, we can culture human endothelial cell and then try to mimic, of course, to a certain degree. Now the flow of different, the impact of different blood flow. And one is we call this this USS unidirectional shear force. Actually, this is a good flow. Yeah, we call laminar flow. Now able to bring up now this expression of UCP2 in this human endothelial cell. By contrast, if you expose human endothelial cell to the disturbed flow or oscillatory shear stress (OSS), and then you see clear reduction in the UCP2 expression, both are in the messenger RNA and the protein level. And another way, of course, even these arches are very small, we're still able to dissect little piece here from the aortic arch, which actually we consider this low shear force, particularly the inner curvature of the aortic arch, and then all the thoracic aorta, which is taken as the location of laminar flow, is a beneficial flow. So you can see, obviously, you can see the difference, different expression of UCP2, and then in this area, for example, the distal flow, you can see in the distal flow, you can see apparent reduction in the expression of UCP2 as compared to the level of UCP2 in thoracic aorta, which is exposed to the laminar flow. Now, because we try to find what the upstream regulator, but in the end, KF2, because also one speaker mentioned KF2, but the KF2 to me is very important and special. This is a master transcriptional factor which controls so many different things. This is one of them, one of them. But here you can see very clearly, now KF2 actually plays a very key role in controlling now the different flow pattern induced change in the UCP2, the expression in human endothelial cells. Now, for example, distal flow, if you induce the suppression of UCP2 expression can be reversed or prevented by the overexpression of KF2 or another way around, for example. In the protein level also, you can see very clearly here. Now, this KF2 actually is a very important regulator of eNOS. eNOS protein, eNOS is the index, and together with UCP2, because our previous study made it linked to the UCP2 to the eNOS activity on the endothelial function, and this all can be demonstrated here. We are more interested in whether or not any drugs, for example, as I mentioned earlier on, because the GLP-1, the receptor agonist able to increase UCP2, or anything else. Now we know a lot better because there are many lots of drugs. Even in statin, this is my favorite compound, and resveratrol, for example, all able to increase now KF2 because they have very similar effect in comparison of the impact of laminar flow now on those changes of genes in human endothelial cells. Now here is very clearly we use two statins and resveratrol, all able to increase UCP2 expression in culture human endothelial cell. And all this effect can be suppressed now after you knock down the KF2 in endothelial cells. Now, whether or not these are useful, UCP2, and then we knock down on the endothelial cell and then induce in the upper E knockout mice on the high-fat diet, for example, to let them to develop the plaques in the aorta. And apparently, now if you knock down the UCP2 selectively, specifically in endothelial cell, actually leading to increased formation of those plaques. And certainly they play a certain role. We also use some other drugs to treat those upper E mice to confirm UCP2 actually is involved in the development of atherosclerosis. This is another way because we're doing this impact of flow, so that's why we use a partial ligation of carotid artery, which because disturbed flow induces formation of plaques rapidly, normally within 2 weeks, for example. And here we can do further the examination, particularly when you test the effect of any drugs, for example. This might be a good model. Instead of upper E mice, normally we need to treat them about 3 to 4 months, for example, before we kill them. Now, this is some of the small summary, but do not have time to go through. But UCP2 is certainly very interesting, but now we move on further and then look at how these things really regulated by disturbed flow. Because laminar flow, we know KF2, disturbed flow is still not so clear about the regulatory mechanism for the suppression of UCP2 in response to disturbed flow. Now, these are probably my summary slides. Mitochondria are the major source of reactive oxygen species, including in the endothelial cells, for example. Now, and this is a major contributor of the oxidative stress in endothelial cell, probably play quite important role under the different disease conditions, for example. That's what we want to do. Because many years ago, we also studied ER stress in endoplasmic reticulum. And we do not really know there's a tool because they have a link, certainly, in transfer calcium, for example, from one source to another source. But we have no any idea whether or not ER stress actually is somehow associated with UCP2 activity. That's something we want to study. And also we try to find more drugs, for example, safer and then more effective to increase UCP2 activity and the expression. Now, this is my slides. I'd like to thank this is actually a photo taken before pandemic. My last photo, that time I was still in the school biomedical science, this university. And now I count the number of my members, actually 9 of them left already. Yeah. So I have fewer people now. I joined the CTU a couple of years ago, but still they work very hard. I'd like to thank those funding, particularly Hong Kong, the Research Grant Council. They're very generous. They give me quite a lot of money. Thank you so much.
Thank you, Professor Huang, for this very interesting talk. Any questions from the floor?
Now I find out what you are doing. So I actually wanted to know whether or not piezo would be involved, linked at all with UCP activity.
Thank you, Kathy. So nice to chat to you. Last week, I cannot remember. Thank you so much, piezo, because now in one study, we exclude the piezo. Because I've been thinking about this calcium permeation pathway, which is controlled by stimulation. And the piezo, there was one, for example, there was first one identified to be mechanosensor, expressed by endothelial cells. And later on, we found there's many other those putative, the mechanosensor. But now I've been discussed with my team whether or not this piezo, which allow calcium influx, properly also sodium, is due to the calcium-sodium influx now induces certain changes, or piezo at the cell, whenever you stimulate it, serve as a, when you have a conformational change, serve as a signaling, and then trigger something unknown inside the cell. So that's why we try very hard and not easy. And also now currently we want to knock out all these, the cell membrane receptor transporting proteins, enzymes, for example, or potentially to be a sensor, and whether or not into our BD, our chamber, flow chamber, to identify which one is more relevant, for example, more important. But I haven't studied, for example, this piezo 1 linked to the UCP2. These are very, very good. And I still believe now anything is a regulatory mechanism involve multiple factors. Yeah. But for one study, we only able to focus on one research area. But this really
So you don't find
interesting.
any change, or maybe you haven't looked yet, or has anybody looked to see if there are any changes in UCP1 activity or mitochondria involvement in the piezo knockout mice? Because there are knockouts, right?
Yes,
for the mice.
yes. But I do not really know this, never be aware of this. But these are quite interesting, because what I've been thinking, talk to my team now, even intracellularly, the certain RS species might have modulatory role on this mechanosensor, but now we're still unable to think the way to study this. Of course, first you need to identify stable, those RS species. Otherwise, it would be difficult. Thank you.
Yeah, really, really nice. I mean, increased UCP2 also reduces ATP generation, obviously. Just wondering whether this enhanced expression beneficial role by reducing ROS is also via this reduction of ATP, some adverse effects on endothelial cells.
No, these are very excellent questions. And even endothelial cell might not consume too much ATP, but still ATP required for the kinase activity, for example, and also those active pump on the cell surface. A little bit probably in the ER, the endoplasmic reticulum for the calcium pump. That actually is quite interesting, but we haven't measured this ATP. But certainly, because now what we did, what we have been doing actually, we try to work on this exercise model, for example, and then get those endothelial cells for further analysis.
Okay, so thank you very much, Professor Huang, for this interesting talk. Because of the time, I think we can discuss more during the lunchtime.
So the next speakers will be Professor Nicola Smart. She is a professor of the cardiovascular science at the Institute of Developmental and Regenerative Medicine, Department of Physiology, Anatomy and Genetics, University of Oxford. So welcome, Professor Smart.
Thank you. Thank you very much. Nobody wants to be the speaker that stands between the audience and lunch. Thank you, Kathy and Bin. It's a wonderful opportunity to be here and join this very exciting meeting. Kathy's already beautifully introduced and reminded us that cardiovascular diseases are the leading cause of death and morbidity worldwide, and predominantly these are caused by coronary artery disease. What's been appreciated more in recent years is the important role that the smooth muscle cells of the artery play in disease. Historically, the focus was on the endothelial cell and on, historically the focus was predominantly on the endothelial cells and the immune response provoked. But increasingly, the limelight is now on the smooth muscle cells. And that's because in a healthy state, they play an important role they express contractile markers and play an important role in maintaining vascular tone and regulating stability. But in disease and in response to growth factors such as platelet-derived growth factor B, they undergo a switch to what's been called a synthetic smooth muscle state. These cells are more proliferative, they're more migratory, and they're resistant to apoptosis. Now, I'm the first to admit that this is an oversimplified umbrella term. It actually describes a heterogeneous population of cells, some of which have been shown to possess osteochondrogenic properties. Some are described as adipocyte-like. Some may resemble mesenchymal stem cells. Some are termed fibromyocyte, fibroblast-like cells. And some adopt a macrophage-like or foam cell fate. As well as a reduction in their contractile properties, they also express an altered matrix, and so they contribute to atherosclerosis by promoting fibrosis and vascular calcification, as well as a loss of vascular tone. And so we can investigate smooth muscle phenotypic switching in a whole range of different ways. Commonly, this is explored in human cell-based models even. We can focus on some implicated genes through GWAS studies. All talks this morning pretty much have highlighted the phenomenal insights that are gained through single-cell omics technologies, and we can leverage high-throughput screening. But our first entry into this area really came from the study of embryonic vascular development. And the rationale here is that if you study the mechanisms that underlie the formation of stable vessels, processes such as smooth muscle differentiation in development, there may be some important insights there to be gained of mechanisms that serve to protect and maintain stable vessels throughout life. Another way to think of this is perhaps the early stages of smooth muscle phenotypic switching may be just a reverse of the process through which mesodermal cells differentiate into smooth muscle cells in the embryo. And so long before single-cell omics was conceived, we were investigating vascular development, and we characterized important roles for the small actin monomer binding protein, thymosin beta-4, and found this to be important for smooth muscle differentiation in a range of vascular beds, including the yolk sac, the systemic vasculature and the aorta, and the coronary vasculature. And the highlight really for this is that in the absence of thymosin beta-4, the embryos lack mature smooth muscle cells, and this is correlated with disrupted TGF-beta signaling. So the 25% of embryos that die during development are those that are unable to maintain TGF-beta signaling in the differentiating mesoderm. Although at the time we had actually no insight into how thymosin beta-4 could impact growth factor signaling, particularly TGF-beta, PGF-beta, the processes that regulate smooth muscle differentiation. So what happens to the 75% of mice that survive the developmental defects? Well, as adults, they're not normal either. Their aortas are enlarged, dilated. They demonstrate elastin defects, elastin fragmentation, and their smooth muscle cells exist in a state that's more synthetic. So this is obvious both histologically, but also in the marker profile they express. What I'm not showing here, this is an old study now, what I'm not showing is that if you, using a conditional knockout approach, if you delay the knockout of thymosin beta-4, allow them to bypass the embryonic defects, you can then establish that actually there is a requirement throughout life in maintenance of healthy vessels. So thymosin beta-4 is protective in the vessels, and actually the best way to appreciate this is through looking in a disease model by single-cell RNA sequencing. In an apoE-null atherosclerosis model, even at baseline, before switching the diet, there are 3 transcriptomically distinct clusters of contractile smooth cells, and already populations that as modulated or synthetic. When you switch the diet to a high-fat diet, you see a progressive transition of contractile cells to the modulated state. And there's a really striking correlation in thymosin beta-4 levels. So the ones that transition to smooth muscle to the synthetic muscle state are the ones with the lowest level of thymosin beta-4. So in fact, thymosin beta-4 is among the top 20 downregulated genes, alongside the likes of smooth muscle heavy chain, smooth muscle alpha actin, And so we see a very striking correlation, positive correlation with contractile genes, shown here, transgelin, and a negative correlation with the synthetic genes, exemplified here by osteopontin. But still, no indication into what mechanism could possibly be driving this at the time. And to cut a very long story short, particularly we wanted to know about the link with growth factor signaling. So to cut a very long story short, we demonstrated that thymosin beta-4 physically interacts with the low-density lipoprotein receptor-related protein 1, LRP1. And the reason this seemed of interest and relevant to us is that LRP1 had been implicated through GWAS with a whole range of different vascular diseases, implicated in aortic aneurysm and coronary artery disease, to name a few. And not unreasonably, the assumption of these studies was that this would relate to dysregulated lipoprotein metabolism, and that's what the name would suggest. But we now, in fact, know that the other smaller LDL receptors are the ones primarily responsible for LDL clearance. And actually, the role for LRP1 was mainly understood through mouse studies, where LRP1 could be specifically deleted from a range of different cell types. And it was actually the smooth muscle specific knockout of LRP1 that gave the greatest phenotype. Those mice are susceptible to abdominal aortic aneurysm and atherosclerosis. And disease in these mice is underpinned by dysregulated PTGFB signaling. So back to our thymosin beta-4 knockout mice, and when we cross these onto disease models, we find that they are highly susceptible not just to atherosclerotic plaque formation, notably in the coronary sinus, but also to aortic aneurysm. Sorry, my screen's gone blank. I'm sure. Okay, the screen's blank as well. I don't know why it just dropped out. Did I do something? Okay, I might just stand back and look at the screen there as long as it still works. Sorry. Yes, and in the most severe cases, in the aortic aneurysm model, there's dilatation, particularly in the aortic arch and descending aorta, and in the most extreme examples, we see rupture and dissection within just a few days of angiotensin II. And what's important to note here is the striking common phenotype between the LRP1 knockout and the thymosin beta-4 knockout. This is just one example. We then went on to describe the mechanisms underlying this, and in the interest of time, I'll just show this in a schematic. So PDGF receptor, LRP1 is a co-receptor for PDGF receptor beta and controls its trafficking through the endocytic pathway. So after activation, LRP1 and PDGF receptor beta are internalized, and from early endosomes, they can either be targeted to the lysosome for degradation or recycled back to the cell surface. And so that obviously controls the sensitivity of smooth muscle cells to PDGFB, which I told you controls their phenotype. And what we found is that thymosin beta-4 was essential for the correct trafficking of LRP1. And so in the absence of thymosin beta-4, disproportionately more PDGF receptor was trafficked back to the cell surface, and therefore the cells were more sensitive to the effects of PDGFB, and that promotes their switching. And so this is just to illustrate to you an example of how the study of developmental vascular development led to some insights into mechanisms that protect against the vasculature in adulthood. And at least in the example of LRP1 and through GWAS studies appear to be relevant in human disease. But how do we know that the insights we gain from animal studies will translate to humans? How do we know that we're studying the most relevant mechanisms and identifying the most tractable targets? We're increasingly aware of the translational gap. It's been mentioned at least once this morning. And so we're now recognizing that human cell-based models are essential to complement the animal studies that we do. And so we sought to model coronary artery disease, or at least the smooth muscle aspects of it, in vitro. We actually tried a range of different systems, trying to increase the complexity through use of co-culture systems, hydrogel embedded spheroids, microfluidic systems. But actually, we found the most robust and system that most accurately reproduces the human coronary artery disease in vitro was a very simple 2D monoculture system treated with the growth factors that drive disease in vitro. So that's the one we've focused on so far. We do hope to improve the others. And so when human primary coronary smooth muscle cells are treated with PDGFB for 24 hours, we see a number of the hallmarks of phenotypic switching. So morphologically, they transition from a very well-aligned spindle morphology to a completely dysregulated rhomboid shape. This is accompanied by downregulation of the key contractile markers, an upregulation of some of the markers associated with the synthetic phenotype. And functionally, they show vastly increased proliferation and migratory capacity. So this was reasonably reassuring, but we wanted to be absolutely sure that we were capturing all of the key events, or most of the key events, that occur in human coronary artery disease in vivo. And so to do this, we went back to one of the very earliest single-cell RNA-seq studies in human. This is quite old now, but it's been extremely well validated. This study took explanted hearts from 4 patients with late severe disease and studied and sequenced coronary arteries at single cell level. If we focus on the cell types of interest to us, we see there's a clear fibromyocyte population that sits between the smooth muscle population and based on not just the proximity of these clusters, but also the transitions that have been described with lineage tracing in mouse, we can infer a transition of these cells from smooth muscle cells to a fibromyocyte state that is sort of on the way to becoming more fibroblast-like. And this is accompanied by a downregulation of all the contractile markers. And we can see through looking not just at these 3 genes, but at hundreds of genes, that our model accurately recaptures the majority of those. And so having ascertained the robustness of our model, we went ahead, and our first experiment really was to just transcriptomically analyze the profile, the transition across a time course by bulk RNA sequencing. So we found a large number of the expected transcriptional changes. Some of those are highlighted here on the volcano plot. But we also identified thousands of novel new genes that we think might be informative. And so if we map all the data back onto the in vivo data set, we find that those that are highest in the control cells align more with the smooth muscle cell cluster, and those that are upregulated in disease align more with the fibroblast cluster. So we're reasonably happy with this data set. But how do you take more than 5,000 different genes and begin to work out where to focus the mechanistic studies? In order to prioritize these, we filtered them using a very clearly defined set of criteria. We wanted there to be consistent gene expression, which we can exemplify using BCAM here as an example. We wanted to see that that their expression profile closely aligns with the expression of contractile markers or contractors of state markers within our model. They also had to show consistent expression profiles fitting with the single cell RNA sequencing data that I've just shown you. We wanted to ensure that their expression aligned with expression in the human cell atlas and the protein atlas. And finally, we wanted to prioritize regulators that had perhaps already been associated and implicated in human disease through GWAS, coronary artery disease GWAS. And then, so having used an unbiased systematic approach, this far. At the very final step, we slightly biased the process because we were particularly interested in transcription factors that might be key regulators of the process. And we also wanted to make sure that we included factors that would both promote the switch and might be protective against it. So our final shortlist of 42 putative regulators we took forward to high throughput screening. And we did this in collaboration with the Novo Nordisk Research Centre in Oxford. And so our assays involved the knockdown of our 42 target genes in the presence or absence of PDGFB for stimulating the switch. And the readouts we used were two complementary assays. One was DrugSeq, which is a transcriptomic assay that allows capture of more than 6,000 transcripts per well of a 384 well plate. And then we did an ELISA, which is a high throughput multiplexed automated assay for analyzing more than 1,000 secreted proteins per well. And again, these were the same wells that were used, the same cells that were used for the DrugSeq, so we can match secretome with transcriptome in each case. So this gave a large data set that describes not only the transition seen with the phenotypic switch, but also how perturbation of our different pathways and different regulators would affect these changes. We used 3 different human donors in our assays, and not surprisingly, the main difference when we separate them by principal component analysis was by the donor. Who thought humans were vastly different to experimental mice? But reassuringly, we see in each and every donor the same, we see a separation of the populations based on PDGF treatment. And when we normalize for donor differences, we have a combined data set where we still see this change induced by PDGFB. Consistent with our hypothesis that we have included here factors that drive the switch and factors that might be protective, we see accordingly that some of the perturbations drive greater expression of the synthetic genes, where others maintain the more contractile state. One of the genes that shows a very strong pro-synthetic phenotype is PROCA. PROCA is a protein that's been primarily associated with preventing blood coagulation. It activates protein C. It's also been implicated in proliferation of a range of different cells, but has yet to be explored in smooth muscle cells. So when we knock down PROCA, we see an increased expression of the contractile proteins, both in the unstimulated and PDGFB treated cells. So the perturbation is protective in keeping with it being a gene that ordinarily drives the phenotypic switch. And so with the vast data set we have at our disposal, the transcriptomics and the secretomics of the pathways affected by PROCA, it was still unclear really which of these pathways we should prioritize for follow-up. What was really helpful here is that a number of the perturbations intersected by affecting some of the same pathways. And the one that really stood out to us was age-range signaling. So age-range signaling, by way of a very brief introduction, drives inflammation and oxidative stress. It's triggered by the glycation of proteins and lipids, and so is typically implicated in a range of metabolic diseases. And although the regulators that we were investigating had not previously been linked to age-range signaling, there's plenty of evidence in the literature for age-range impacting smooth muscle phenotypic switching, diabetes-induced vascular disease, and so on. So this really piqued our interest and gave us a focus now. We are following up on these studies, trying to understand how these pathways impact age-range signaling and whether this could be a meaningful target for protection against vascular disease. I'm afraid I don't really have those data yet to share, but maybe next time. It remains. Oh, sorry. So this, in summary, I've shown how studying human-based models, investigating GWAS, the study of embryonic vessel development, single-cell omics, high-throughput screening allows for the study of a very important disease mechanism. Then it remains for me to thank those in my group and our collaborators who carried out on this side the LRP1 thymus protocol study, and on this side the human cell smooth muscle cell model. And finally, thank you for your attention.
Thank you very much, Professor Smart, for this interesting talk. Any questions?
Hi, Nicola. Very beautiful work. I'm asking a question about the nature of those synthetic smooth muscle cells expressing thymosin beta 4. Do these cells also express some senescence markers? Because previous studies show that these contractile smooth muscle cells become synthetic smooth muscle cells. They will become macrophage-like cell or fibroblast in the atherosclerotic plaque when they migrate into this plaque and express some senescence markers. So it is the same similar population that express SASP.
Yes, we did. This was a while ago now. I don't think we looked specifically at senescence markers, but they certainly express very high levels of inflammatory markers. In fact, one of the even the contractile clusters expresses a much higher level of inflammatory markers than the other two clusters. We could easily go back and look at senescence markers. They certainly weren't within the top affected genes. But I dare say you're right.
Really nice, Nicola. I was wondering, do you actually also see spontaneous plaque formations just in the thymus beta 4 when you have more synthetic cells? I mean, we did a similar experiment years ago when we knocked out microRNAs 143, 145, and we saw their spontaneous plaque formation, but without inflammatory cells, and without actually senescence. Just this enhanced proliferation was of smooth muscle cells was sufficient to drive their atypical, I have to say, plaque formation.
So certainly in a number of older thymus beta 4 knockout mice, we detected a few plaques. We didn't really power the study to go through systematically looking for them. It's almost like looking for a needle in a haystack. But they certainly had dilated aortas, other vascular defects.
Actually, we also saw that at rather atypical locations. They're mostly abdominal aorta, not in the aortic arch. So still wondering actually how this works, but we didn't follow that up.
No.
Any short questions, if any? Okay, if not, thank you very much. Thank you very much. So thank you once again for all of your attention to this section.
Thank you, moderators and our speakers. May I invite all the moderators and speakers for this session to come on stage for a group photo. So they are Professor Lee, Professor Zhang, Professor Dimler, Professor Augustine, Professor Huang Yu, and also Professor Smart. Let's give them a round of applause. Thank you for the groundbreaking insight and the stimulation. So let's take a group photo to capture the moment together. So let's look at our official photographer. Let's slightly walk to the side, towards this side. Yes. Yeah, let's make one step forward to this side. Okay, thank you. Okay, thumbs up. Perfect. Thank you so much. Please take a seat. Thank you so much to our distinguished speakers and moderators for their insightful presentations and engaging discussions. Ladies and gentlemen, now we have come to the lunch break. The organizers has provided light lunch to the registered participants, and you may enjoy it in the reception hall over there. For our guests who are invited to the luncheon, please follow the instruction from our helpers to the restaurant, during which we will go through the beautiful CUHK campus. And in addition, we invite you to visit our sponsors booth located just outside the venue to learn more about their innovative work and contributions. You will also find poster presentation area nearby featuring 15 outstanding posters showcasing research findings and project highlights. We encourage you to explore these displays and connect with the presenters. We'll see you soon again at 1:45. 1:45 with different keynote speakers with their insights on different topics. See you soon at 1:45. Thank you very much.